And welcome to our third annual speaker series, Engineering for Life. So our focus today will be on how scientists are applying engineering to biology in order to develop groundbreaking technologies. And then our speakers will be presenting on a wide variety of bioengineering topics. And currently, I think we have two speakers present, and all three. Okay. And then we'll have a 10-minute intermission, and then we'll round up this evening with our okay. This is not relevant because we have all three speakers. All right, so before we begin, hopefully everyone like put aside their phones or put it away. You don't take any photos or videos, and if you have any questions, please save it for the end, in which we will have a Q&A for the speakers after this. And then so our first speaker today will be Professor Endy. Dr. Endy helps new undergraduate majors in bioengineering at MIT and Stanford, where he runs a synthetic biology research lab. Dr. Endy also co-founded the IJIP competition, engineering Olympics, if you could say, that enables over 60,000 students globally. And then the Esquire magazine also named Dr. Endy as one of the 75 most influential people of the 21st century. And with that, I would like to hand it over to Dr. Endy.
Thank you, everybody, for being here. I just might have to test from the story before teaching on campus, so it's comfortable. Would it be okay if I bring my mask down? Thank you. Also, happy to take questions. So I wanted to bring to you a dream, and the dream is one in which 10 million people on this planet flourish in partnership with all of our And I feel that that dream is possible, and that to get to that dream, we have to figure out how to partner with life to enable civilization scale flourishing with I believe that to be true within a generation. I believe it to be unlikely to happen spontaneously by default, that it would require and cooperation. So that's my claim at the outset, and I'm hoping to introduce you to why I believe that and take your questions along the way. Does that sound okay? This is something I want to start with, an acknowledgement, incredible film by Jesse Rothman called Growing Is Forever. It teaches me that life and nature, well, life at least, pre-existed before Homo sapiens. And although we inherited, at least we feel that way sometimes, we can't take it for granted. So I just sort of want to give this as an opening acknowledgement of the relationship between humans, Homo sapiens, and the rest of life. What I'm going to do next real quick is offer different perspectives on how people relate to biology. So one way is through food, through the bounty of a harvest, whether it's a First Nations harvest or industrial agricultural harvest. This has long mattered, always mattered to us as a set of societies. The science of biology, learning how biology works, learning how life works, has long mattered, not as long at all. So taking examples from the last century, Barbara McClintock and colleagues discovered transposons or mobile genetic elements. If you think about maize of corn that has different colors on the kernels, that's related to these hopping genetic elements that Barbara and colleagues figured out. More recently, we've seen the fourth generation of DNA editing tools here, CRISPR. How many folks have heard of CRISPR? How many folks have heard of the woman on the right, the bottom? That's her. Anybody know her name? Jennifer Doubt. And just for fun, to help you remember, she spells her last name D-O-U-T-N-A. So I want you to imagine those letters on the license plate if you saw that car going down the street. D-O-U-T-N-A. What would that say? It's not true. What's the chance that a professor at Berkeley was recognized with the Nobel Prize for the co-discovery of CRISPR and its usefulness as a last name that spells out. The technology of biology has mattered in this molecular way going back to the mid-1970s. Shown on the top left of the screen is a United States patent granted in 1980, issued to Stanford University. And it's the first patent for genetic engineering, making chimeric molecules of DNA. If you have two molecules of DNA, you could use the enzyme to stitch them together. This gets birth to the modern era of genetic engineering. Starting around that time, literally in 1980, we see these tools applied to create goods and services that define so-called bioeconomy. And this bar graph indicates that there's three big sectors of the bioeconomy that genetic technologies get deployed to do so. One is in agriculture and food. The second is in medicines, making medicines, and then the third is in materials, like carbon fibers or other sorts of things we need as an industrial and legal society. What's interesting about this plot is the y-axis is money, dollars per year in revenue. So if you've heard the gross domestic product, you could ask what is the genetically engineered domestic product. And you can look at the shape of the curve and you can see that it's been going up for the last 40 years. The total volume of economic goods and services doubles about every 7 years. On the back of genetic technology. Not without controversy, but just to give you a sort of macroeconomic context. What does this look like practically? The top left image, the gray, bluish objects, those are Saccharomyces cerevisiae, brewer's yeast, or baker's yeast, for bread making or beer making. These yeasts have been engineered. When they take in glucose, they don't make ethanol and carbon dioxide. They make, in this example, farnesene, this oily yellow substance that's an industrial intermediate that would normally be derived from a barrel of petroleum. So that's what we mean by an industrial product. This was done in Emeryville, California. Before that, recombinant DNA or genetic engineering was used to take genes, for example, encoding the human insulin molecule, put that into microorganisms such that instead of collecting insulin from slaughtered pig organs, you could have a fermentation process in which the microorganism would make the same protein or a better protein, the human insulin protein, from a broth of bacteria. And verify that and use that for treating diabetes. That gives birth to genetic tech and literally. The controversy today is why is this so expensive? Because the cost of making the covalent bonds defining the protein is approximately free. Separate topic, but relevant. Bottom left, that's a papaya. But this papaya is infected with a virus called the ring spot virus. That's Gonzalez and colleagues working in Cornell, but also Hawaii, figured out how to genetically engineer papaya to make it immune to the ring spot virus. They did this before big agriculture got a hold of genetic technologies. This is more of a local biotechnology application that salvaged the papaya agricultural process in parts of Hawaii. Again, not without controversy today, but an example of how a local biotechnology could be advanced. On the right, those materials look like cow hides, animal hides, but that's not what they are. They're mushroom skin. And so it turns out you can take the organisms in the forest that are the primary decomposers of trees, what they call that, the wood buckets. And if you give them wood chips and inoculate them with a liquid culture of these mushrooms, that within a few days, the wood chips will grow into a spongy mass. And then within a few weeks, the surface, think of it like a giant lasagna tray full of woody stuff. The surface of that lasagna tray will differentiate into a mushroom skin and peel off and put through a tanning-like process to make a hide, or a mushroom hide. So you see luxury goods brands like Hermes and Stella McCartney beginning to make shoes and handbags and whatnot using mushroom leather because it's animal-free, cruelty-free, carbon neutral, and no mammals. So a company like MicroWorks or Primitive and Paul, two of those three companies are in the Bay Area, pioneering this one. But be unfair to tell you that the productive things that are happening, because the context is, as you well know, not totally great. One of the things that's happening is extinction is raging. To the extent that we're tracking loss of biodiversity in nature, and I'm sure you know better than me, my understanding is we've only ever really described less than 10% of the species on Earth. But for the species we are tracking, over my entire lifetime, 50 years now, as the human population has doubled from 3.5 billion to 7+ billion people, the natural biodiversity of the seas have dropped by a factor of 2. So twice as many Homo sapiens. The biology we know about half of as much as anything else. So that's not great. Why? Many reasons, but one of the reasons is changes in land use. So the top right image shows an oil palm plantation. And if you regularize how the land is used to make oil palm growth situations to make palm oil, the more diverse jungle gets displaced. So the animals that live in that natural environment that's more diverse don't have a home. If we industrialize agriculture with current nitrogen and phosphorus runoff in the natural estuaries, you get algal blooms and other types of knock-on effects in that way. And then, of course, for a pandemic still. The word pandemic literally means all people, right? And the horrible experience reported by the New York Times last week was 1 million deaths just in the United States that we know. So I'd be doing everybody a disservice if I appear here as a bioengineer and say, biology is doing great. Right? It's amazing possibilities, but we're navigating a number of aspects which are crises. This is a photograph of the fire, the CZI electric fire on the second ridge. Right? And so I used to, with that opening movie from Jesse, growing is forever. Now I'm not so sure. Is growing forever? I'm not so sure. When I drive up and down the 280, certain parts of this side of the ridge, the forest looks brown. I think it's 50-50, it's going to burn in a second before my kids have a chance of graduating from high school. Right, so that's terrible. I don't mean to be scary, but that's not the thing. So I don't take this for granted. Keep going. So I want to pivot and contextualize bioengineering in a much broader context. I hope that some of you, or all of you, learned about Martin Luther King Jr. and maybe you recognize this photo from his most famous speech. I want to remind you what the speech was not called. He didn't give a speech saying, I have a plan, or I have a vaccine, or a treatment. The speech was not, I have a bioeconomy. The speech and the genius of it is that it's about a dream. It's about a story about a future. And by sharing a story about a future, it gives you the chance of organizing people to work together towards that future, to co-labor towards the future. So I want to talk a little bit more about dreams, because when I encounter bioengineering, as much as I'm encountering science, engineering, and laboratories and learning, I'm also encountering storytelling. I'm encountering culture and politics and the structure of the narrative, how you choose to tell stories. That ends up being as important and perhaps more meaningful than the hard natural sciences and engineering. Nothing against that stuff. So let me give you a quick tutorial on how we think about dreams. We think of dreams, since I'm a nerd, I'm going to think of a dream as a tool. And it's a tool that I can use to make sure that I'm happening to things, so you can make sure that you're happening to things. Is an emerging technology happening to you? Is AI happening to you? Or are you happening to AI, artificial intelligence? Is the future happening to you? Or are you happening to the future? Is the future determined by outside forces beyond your control? Or is it something you can navigate with individual agency and bring other people together? So dreams are tools that you can choose to use if you want to position yourself with respect to your relationship to the future. And I just want to spend a moment further to illustrate how you can make choices in terms of how you deploy dreams. So everybody knows as an actor, nightmares are very powerful tools for organizing yourself out. The hidden cost of using a nightmare dream is is people have to believe the nightmare is coming true or already here before you get into action. Example I can give this is Vice President Al Gore and his famous documentary called very powerful, impressive documentary. It's telling a story about the future, the inconvenient truth. This is a story about climate change. It's a nightmare story. It's very powerful, but it comes with a hidden cost. By adopting the negative framework, we will not realize collective action until a sufficient number of people believe the bad thing is happening and they're experiencing. So it's a valid way of organizing yourself with others. So stated differently, this will sound weird. It's not the weirdest thing I'm about to say. Climate change as a narrative choice makes climate emergency inevitable. Because only when people believe the climate emergency is here will we get sufficient collective action to attempt to fix it. Martin Luther King's dream was positive. Right? We have other examples of positive dreams. Positive dreams are harder to believe in and organize, but if you can do it, the benefit is you get collective action in advance of the reality. Right? People will co-labor to make the impossible thing real. To follow projects, getting to the moon and back is another example of a positive dream. So here's a strange bit of mind. What if we instead reframe climate change as climate opportunity? Oh my gosh, there's so much carbon in the air. And then we get climate awesome. Now I bet those words sound hard. Not really that way. Right? That's just because the structure of the narrative has been multiplied by -1. There's a negative narrative. Not to dismiss the dire nature of what's going on at the time, but just to give you a dream this tool. So let's bring that back to biology and bioengineering. Just like we had astronauts, I wish to imagine people as bionauts heading into a biotic future where within a human generation by 2050 we have a flourishing civilization on this planet. Let's apply the dream tool. What are our dreams? What are our positive dreams other than biology? I'm going to use facts on our side and an example on the right side. I have courage that you can figure this out for yourself. That anybody knows what's shown on the side. One bioengineering majors, like I said. So my undergraduate major in college was civil engineering. My internship was construction management, working for Amtrak, doing excavation contracts on the East Coast. On the first being for that. But I didn't have a bioengineer because that's what it's building, it's growing. Long story, but that's how I am. So as a civil engineering student, you see what I like about the civil. It's a double-decker suspension. From the roots of the rubber plant. It fixes carbon over time. It's handed down from one generation to the next. If you take care of it, we'll get stronger. The opposite of all records. So how do I think about biology as an engineer? It's all over the place. It's powered mostly by photosynthesis, and we can quantify that. The photosynthesis harvested by nature is about 90 terawatts of energy. A terawatt might not make a lot of sense. So to give you a frame of reference, all of human civilization on average at any instant consumes 20 terawatts of energy. Nature is powered by 90 terawatts of photosynthesis. Humanity is crawling 20 terawatts. You all know that 90 is 4.5 times bigger than 20. So right there, on your napkin map, we should be able to, once biology do its power, mix stuff, organize these molecules and materials and cells and creatures. So right there, since 90 is 4.5 times 20, we should be able to make enough stuff for 10 billion people. We just get bigger credit. There's other things that we know are unique about biology. It reproduces, heals, and evolves. The ethical and cultural ramifications are off the charts. Where did that virus come from? From the nature, from the lab, the political tension around that ambiguity, the ramifications of anything to do with bioengineering in any way you can imagine the field. And so we could tweak the facts on the left, like welcoming anybody who gives me an upgrade on these facts. The question is, what do we want to fight? Given bioengineering appearing as an engineering discipline, what's our dream? I don't know what it should be for you, but I'll tell you what I got. And I'm just struggling to come up with it. Let's enable the civilization to provision for itself. Let's stabilize and recover natural biodiversity. Let's take effect. We have to make infectious diseases obsolete. We should make other diseases obsolete too, like cancer. But we definitely have to make infectious diseases obsolete, not just because of natural pandemics, but you know, what if something actually made a pathogen? I'd like my kids to grow up and their kids to grow up and their kids to grow up in a world where they are citizens of a bio future, as opposed to consumers of that future or subjects or objects in that future. What is the relationship between these technical capacities in our societies, our democracies? By the way, you take something apart like a bicycle and you think you understand a bicycle, but if you put those pieces back together and you're working a bicycle, you definitely have like a higher confidence in understanding the bicycle. And same thing's going on with the science of biology. Science and biology historically has mostly been observational, and then take the things apart and look at the molecules, like taking the bicycle apart. But synthesis in biology, which is just now spinning up, let's just put biology back together and see what happens. So laboring to whatever the goals are, very fundamental is to understand that much better. Building. Again, it's not a prescription. I just want to give you an existence proof that you could come up with your own dreams. I encourage you to come up with your own positive dreams for the future of our relationship with the world. Does that make sense? Like I mean it. I wish we could do this. Okay. How do you work for time? For me. How much longer do you want me to?
Whatever you,
I can't hear you.
whatever you need.
That's a very different statement to make to a professor. I need a number.
10 to 15.
10 or 15?
Yeah.
Is that what you said?
Yeah.
Thank you. The other, even though we, by the way, that number, like how big is the bioeconomy? About $1 trillion a year. That's correct. That's about 5% of the U.S. economy. And you know that the U.S. economy numbers inflated by all this financial transaction stuff. So it's like your real economy or add-ons. Right? So 5% of the economy is a massive number. Even though that's true now, most of what we do with biotech is not much. And so I just want to give you an example. These are tobacco plants. Again, they have been genetically engineered to express a light-producing cassette taken from a wood fungus, a viable mess of processing in the forest. So all plants that we know, right, take in light energy and then chemical energy and store that and then use it. These plants do that, but they also run in reverse and give stored chemical energy and convert that into photons. So there you go. Right? So they're called glowing plants. Maybe for the first time in the last, recorded for the first time, I think it was December of 2019. We have them in the lab just to understand them. They were made in Russia originally and in Russia, and over the holiday break, we brought some home and we're using them as night lights. So you don't have to plug in your night light, you have to water it. We had glowing plants in the natural ecosystem. I'm sure that will be interesting. We might have to look into for sure, which is like we have it with how things navigate. I just don't know. What would you do with glowing plants? What else could you do? Another contextual thing, you are privileged to, I would say, we all are, but you especially to be in a moment where how people decide to organize things to explore and enable or oppose biotechnologies is very much up for grabs. A lot of the infrastructure, cultural relationships that you define within this decade and the next decade. The people who invented genetic engineering a generation ago, it literally was a generation ago. And I'm of an in-between generation. You know, I'm not 70 years old, I'm not 20 years old. And so you are part of a second generation encountering these technologies and have a lot of agency in terms of being able to choose, you know, what lessons from the past to carry forward and what new ways of engaging. So to give you an example of this, totally esoteric tweet. This is a tweet by Dr. Eric Schmidt. Eric was an engineer working at Sun Microsystems and then got recruited to be the CEO of Google when Sergey and Larry were very young. And he's since retired. And he's thinking about this frontier of bioengineering called synthetic biology, which is what I look at. It's not his expertise, just observe it as a techno-strategist. This is about to undergo, this area of research is about to undergo a Cambrian explosion. How many people know about the Cambrian explosion? What was it? So it wasn't a physical explosion, it was a dramatic increase in biodiversity that happened a while ago. How long did it take? One period. So what does Dr. Schmidt mean by this statement? If the Cambrian explosion, this significant increase in the forms of creatures on Earth, that took about 20 million years to play out in the fossil record, best we can understand. Does he mean that this frontier of biotechnology will take 20 million years to play out? Underlying forms defining how these procedures and industries take shape, those are getting set up now. So that's what I mean by your discussion. How many people here have ever heard of the word synthetic biology? I just want to give everybody a quick on-ramp into this space. And the best way to do that is to talk about the synthesis of DNA. How many people have heard of DNA? Right. So normally when you hear of DNA, it's how it exists in nature, it comes from our parents, that's where our machine is. Right? When we think about this process by which it's copied replication. But imagine having a synthesizer for you, printer. A musical synthesizer has 88 keys, let's say, and you are testing the keys, put information into the machine, the music comes out of the machine. With that in mind, imagine a keyboard that has 4 keys, A, T, C, G, the basis of DNA. And depending on how you play the keys, the machine will physically assemble that strand of DNA from scratch. So that's what I mean by a DNA synthesizer. Go from a specification of a sequence to a string of atoms manifesting that molecule of DNA. So if I put in TAA, TA, CTA, CTCA, CTA, TA, GGA into my DNA synthesizer, the DNA synthesizer is going to physically assemble that strand of DNA from scratch, not copy it from some other molecule. Does that make sense? When I first started teaching at MIT in 2003, the cost of pressing 1 key on the DNA printer was $4 a key price. Now, so if a gene is 1,000 letters, it's $4,000 a gene. Protein gene or something like that. Now we've seen the price drop. We've got the price down to about 7 cents to 4 cents a key price. So building a gene is about 40 bucks. I'm hopelessly biased. I think this is the most important part of technology, if you will, for this section. Accelerated wafer fabrication is kind of important for Silicon Valley, but DNA print, because biology is way more important than hardware. I'm biased. I don't care about hardware, more than hardware. Nothing is hardware. And DNA fabrication is the most important thing. I'm going to give you an example of why this might be something you agree with. The SARS-CoV-2 pandemic first started spreading in December 19. The first cases in Europe were reported in France in February of 2020. Switzerland has its first case come in. An infected person travels across the Italian border, and that case is reported in Switzerland in, I think, in 20th of February. Yes, in 2020. But it turns out that's not how SARS-CoV-2 first got to Switzerland. first time SARS-CoV-2 got to Switzerland is a scientist in Shanghai, China, sequenced the genome of the virus, uploaded the sequence to the internet as a string of letters or bits. Researchers in the Swiss capital downloaded the sequence of letters. They resynthesized the DNA template for the SARS-CoV-2 genome. They then did a transcription reaction to make an RNA molecule, and they transfected, put that RNA molecule back into cells in a laboratory dish, and those cells got infected, and live virus was produced. And all of this was demonstrated in completely 13 days before the first infected person got to Switzerland. The internet, in other words, transmitted the virus to Switzerland faster than the pandemic. DNA reading goes from atoms to bits. Right? DNA printing or writing goes from synthesis, goes from bits to atoms. Does that make sense? You get that now. Why? Is it good or bad that somebody's able to get the virus to switch over 13 days before the pandemic? How many people say good? How many people say bad? How many people say no idea? I think all responses are valid. It's up for grabs. We could have public health biodefenses that operate at the speed of light through the internet, or we could have distributed bioterrorism. But anybody anywhere can make any happen. So I would prefer, and what we do is consistent direction, the former, public health security biodefense that's like to be enabled. But that's literally not what's up for grabs. It's not given for grabs. Because I can synthesize whatever DNA I want, I can do strange things. Here's a fun example from researchers in Israel. They took a 3D printed bunny rabbit and they took the computer file specifying how the 3D printer should make the bunny. And because that computer file is zeros and ones, I can map zeros and ones to ATCG. Right? Zeros and ones is just 2 bits per number. ATCG can store, zeros and ones is 1 bit per number, 1 bit per digit is 0 or 1. ATCG can store 2 bits per base. And so I can take an arbitrary digital file in zeros and ones and map it to a DNA sequence. And then I can synthesize the DNA encoded out of biological object, which is the digital file. I can take a book, I can write the book into DNA. And now my library is a big analog object. But I've got a little test tube with a whole bunch of synthetic DNA encoding the information specifying the books. Or in this case, what they did was they encoded the information specifying the files for programming the 3D printer. And so now they have synthetic DNA that is in essence the genome, the digital genome for the 3D printing of this bunny rabbit. They mix that DNA into the bunny rabbit polymer itself. And so now once you print the bunny rabbit, you can take a little snip of its ear, extract and recover the DNA, sequence it, read it out, recover the computer file, and reprogram the 3D printers to print more bunny rabbits, plastic bunny rabbits. So they've just put a DNA genome in a plastic bag, and the instructions are going through the 3D. You can take all the information in this library, all the information to rebuild human civilization, turn it into a digital file, synthesize the DNA encoding that, put that DNA in the handle of your refrigerator. So that embedded in the plastic of the refrigerator is all the information needed to rebuild civilization. You put arbitrary information through synthetic DNA and arbitrary objects. Does that kind of make sense? So when I first started hearing about this type of stuff, I thought it was just like a joke. But it turns out that the archivists, the librarians of Congress, people who are thinking about storing information for 500 years, are very excited about this DNA-based data storage form. It has nothing to do with living biotechnology, it's just a archival data storage. And the reason they're most interested in it is when you think about a digital format, the hardest thing to think about 5 years from now, or 50 years from now, or 500 years from now, is how you're going to read out the format. Like 8-track tapes or the CDs or the DVDs, like the player technology keeps getting outdated. But by moving to DNA format, they have confidence that if people are still around in 500 years because we collectively love ourselves, we'll still be able to dig out our DNA, and so we'll still have DNA readers. So they have confidence that the DNA data format will survive over the centuries in terms of computer technology. Crazy interesting. Okay. 5 minutes? Okay. So I grew up in a world that's called an industrial world. This object, designed in California, made in China, shipped through shipping containers and consumed as a consumer. And it's centralized manufacturing, centralized organization of people and capital that cause things to get there. Biology turns out to be the opposite of this. The leaves on the tree grow where the tree is. They don't come from a factory that makes leaves and they don't get shipped in pallets and then taped to the twigs. Right? The photons arise with biology, it's the carbon dioxide, the water arise. So biology teaches us that all atoms are rollers by definition. And so we have the capacity to enable fully distributed manufacturing. Anything that I can grow, I should be able to enable the growth of that thing wherever I can operate biology. I don't know that this will displace industrial centralized manufacturing, but it'll certainly complement. Let me give you some examples. So this is the work of my wife, Dr. Christina Schmolka. So just to admit, I'm hopelessly biased. What she and her team have done is take baker's yeast, again, for baking or brewing, and reprogram its metabolism to implement bioengineered chemistry so that when glucose comes in this example, the molecule around 5 o'clock on the bottom of that cartoon of the yeast in the left, that molecule is called scopolamine. If you ever have a motion sickness patch, that's the same molecule. Now, scopolamine is normally found in the plant from the nightshade family of plants. And plants, as you know, are rooted. They can't run away from predators. And so they've been selected by evolution and all sorts of interesting molecules that modulate the behavior of insects and birds and animals. And so that's why there's a lot of bioactive chemicals found in plants. Scopolamine, again, being one of these examples, all that chemistry we see in this cartoon we sell is all bioengineered. It's not the natural metabolism. 30 or more enzymes have been bioengineered in this microbe so that when it's fed with glucose, instead of making ethanol, it makes scopolamine. You can do this for morphine. You can do this for 300 different essential medicines. Our drug supplies are based on plants, going to depend on agriculture where we grow plants. So scopolamine is grown in big farms in Australia, right, which were nearby the recent fires and so on and so forth. The brewing process, you can operate anywhere you can run a brewing process, anywhere you can have a ferment. So serotonin, language, where we think. So anything we can turn into a growing process, we have a chance of provisioning it locally through a distributed, debuildized supply chain with resilience around the manufacturing. Right there, if you understand these words even a little bit, you can see the geopolitics of this, the politics of this, the economic aspects of this, the cultural aspects of this are going to be in the I'm going to jump around. I'm going to do one more tweak just to get you the picture. How many people have heard of salmonella? Okay. And if you know it can give you a elementary distress or worse, right? Cause problems with your gut and stuff. Did you know Roy Curtis III and his team bioengineered salmonella to not cause disease, but to instead deliver DNA for vaccinating, in their example, mice against influenza or flu? Imagine having a vaccine that's just a spoonful of a probiotic. No needles involved. One of the problems with the COVID-19 vaccine is that they're not fully biological. Yes, they're synthetic RNA vaccines, but they're packaged up in lipid nanoparticles and then they're injected in plastic syringes and sterile stainless steel needles. And so manufacturing anything that's not biological doesn't grow the product. So it's hard to scale up manufacturing, it's hard to distribute the product. But how easy, relatively, would it be to make 1 trillion doses of salmonella? It's easy. Just keep growing. Why don't we have that yet? weird. It's not a tech, there are technical challenges, but the real reason we don't have it yet is we're not culturally ready to explore and develop and make reliable, fully living vaccines. This has only really been applied to mice and chickens. So let me give you another example. This is an example that Stanford undergraduates explored a couple of years ago. What's a sinus infection? I got microbes going crazy up in my nose, up in my face. Well, what if instead you could have friendly parental microorganisms that live in your sinuses, and they're just naturally uptaking environmental nucleic acids that are nearby in your sinuses. If nothing's happening, they don't really do anything. But let's imagine I bring in a nucleic acid that's a food gene, a segment of the food gene proposed. Well, then maybe I should detect that with a CRISPR molecule and cause the production of an enzyme that changes the color of your mucus. So what comes out of your nose when you're infected with flu is bright orange. And why do I have to go somewhere else to tell me what's happening with biology where I am? Can't I just program the biology to tell me? So instead of diagnostics, it can just be surveillance knowledge. If I'm infected with a coronavirus strain, a different sequence will be recognized and my mucus will turn purple. So to the 5-year-old and 9-year-old we live with, we call this the Purple Booger Project. That feels a lot better to me, so long as we can do it safely. Interesting about these examples, they're examples of fully biological technologies, as opposed to hybrid technologies where you're putting the biology in a fermenter, where you're putting the biology inside a syringe or something like that. So just want to tweak your interest in what could be a fully realized bioengineering. Okay, and so to wrap up, I want to just end with a political question, but not partisan, more in the way relevant to democracy and citizenship. What I've shown here are two artifacts. On the left in the text is a letter that Thomas Jefferson wrote to John Adams in October 1813. And in this letter, he's talking about how if people have access to fertile land, then they will be able to have a good life and eventually retire. And because of that, they'll be interested in law and order. Now, Jefferson is an imperfect person to talk this way, right? But he is very smart when it comes to democracy. If you abstract these words, what he's saying is the option of accessing the means of production is the way of securing a good life. And if you can secure a good life, then you'll be interested in law and order. If you know that I can go over there and secure a good life, then you're less likely to attempt to oppress me. So I'm less likely to actually have to go over there and remake my good life. And if it's mutual, then you're less likely to oppress each other. I know it's a little bit abstract, but it's a genius. To give you the same argument, but from a more modern era, the bottom right is a very interesting book by Dr. Alondra Nelson. Alondra normally teaches at Princeton in New Jersey, but she's on leave and is the director of the Office of Science, Technology, and Policy for President Biden. In this book, she's called Body and Soul. She's talking about the Black Panther Party and how they were implementing public health clinics in the parking lots of Oakland, help members of their community do genomic testing to see who was carrying sickle cell markers for sickle cell disease, because they could not trust the medical establishment at the time to not harm them as patients. That's an example of how the option of accessing an essential capacity can protect you from the risk of political oppression. What does this have to do with bioengineering? Well, if bioengineering is operating at the intersection of joules, bits, and atoms, 90 terawatts of energy, DNA synthesis and printing of any bioprogram you want, and manufacturing on a distributed global scale, it begs the question, who's going to be in charge of this stuff? Who's going to have the option of accessing it? And how we decide to answer those questions will determine whether or not this future tilts towards authoritarianism or liberal democracy, lowercase liberal democracy. Let's turn this into a more nerdy question. How many people should have the option of learning to read and write? Well, everybody, basic education. How many people should have the option of learning to read and write Python or some computer programming language? Why not everybody? Computer science for all. How many people should have the option of learning to read and write DNA? When I hear this question forward, more often than not, I get it wrong for the first two versions of the question, but when I get to this one, it's like, not so sure. It sounds this. For reason. Again, I'm not going to prescribe the answer, but I definitely want to work you up to give you a comment in these types of puzzles and questions associated with this science. So, last one we can talk about or say, I'll just end on the slide again. What are or your positive traits other than biology? Thank you very much. Anybody have any questions? Or strong opinions or objections? Yes. Have you talked to any politicians? The question is, how can we get politicians to prioritize things like climate change, climate opportunity, bioopportunity, like climate, or storm technology, like climate? And my immediate response, honestly, is have you talked to any politicians? So my first response is talk to politicians. But just human beings too, believe it or not. And then my second response, which is easier said than done, is run for office. But the simpler thing is to start by talking to a politician. For example, two weeks ago Monday, I was in Washington, D.C. I flew there to have dinner with two politicians. I paid for that trip. And that we just had a private dinner in a restaurant. So a little table in the kitchen of a little restaurant in Washington, D.C. And there was one person who flew down from Boston, and I flew into California. And the four of us had a conversation. We had a conversation about this conversation we're at the same day. I was going there as mostly an educator. They were teaching me as much as I was teaching that. They were also teaching me about actually my conversation. They're different people in terms of political and cultural values. There's also a lot of real-time politics topics totally unrelated to biology. But because of that conversation, I did not say that I had a conversation with a politician. And I understand what they know, what they don't know, what they worry about, what they don't worry about, and that gives me a chance of sending text messages, WhatsApp, and Signal, whatever, with members of the government. I can guarantee you, if you don't talk to people, they won't listen. If you do talk to people, they probably won't listen. But if you listen to people and find ways to engage in dialogue, then you can get symmetry. You have a chance to get symmetry by having an actual decision. But the other thing to do, I wouldn't shy away from it, is get elected. Get elected. Organize a movement. Share a positive trait. Easier said than done, but I know you can do it. You know, and then anybody in this room can do it. They choose to. I'll say one more thing. It is true that money has an outsized influence in approximately everything. Because how we operate a society. And so one of the things I've experienced is as I get older, my students, no longer students, but go out and work in the real world. Some of them start companies. including really big companies. And as soon as that happens, it becomes easier to have certain types of conversations. So I want to acknowledge that the business sector in our society is in a very important way of bringing forward political conversations. Does that make sense? Yeah. But it's also like good to have options. Like as an academic, I'm more trusted and neutral, right? Capitalism, right? Commercial for-profit example. So it's other things like it's good to have different types of people in patients and these kinds. But if, you know, like stated differently, coming back to you, if I were less than 20 years old today, or come up on 20 years old today, I would insist that there's a positive dream for the world to be like in 2050. What needs to be true for things to be awesome in 2050? And if you and your colleagues start explicitly declaring what that should be, then there's a chance of collective co-laboring towards that goal. Greta, have you heard of this person, Greta, talking about climate? She's a hero, but one of the pitfalls she's landed in is the negative trend, which she's inherited. She's not wrong that there are crises, and I want to acknowledge that. But there's a limit to how much collective action can be realized through a negative trend. What's the place we're trying to get to? So in so many ways, not just get on. I'm with you. I will do anything I can to help anybody here on their struggle. And we'll make it. Like a lot of people in my community, in my generation, feel like our job is to give you infrastructure, equipment, and capacities so that you have a chance to make it awesome, just from a generational care forward. Like the movement is going to come from your age bracket. That's like I never heard you told wrong. It's just like telling you. That's like yeah. Yes, here. Sorry. How long to synthesize a strand of DNA? How long does it take to synthesize a strand of DNA? So right now, most of the synthesis makes fragments that are 100 or 200 letters long. And that's a process that can run over several hours. But it's a process that can also be run in parallel. So you can make 100,000 different strands, each 100 letters long. Does that make sense? All right, so let's say that's going to take you an afternoon or overnight run. Once you have those, you have to figure out how to stitch them together. That's going to add another 2 hours. One of the best companies in the world for doing this is Agilent, the offshoot of Hewlett Packard, so that inkjet printheads for inkjet printers can also dispense the chemicals for making DNA. So they have high throughput printers in Santa Clara. Practically, if you order this as a service, it might take you a couple weeks to get 1,000 or 10,000 base per construct. So when people did that work, it took them a couple of weeks for when they have the information to when they have the DNA. But that was still faster than just 2 weeks in the pandemic. The DNA print technology is very, very immature compared to what it could be. When you think about how fast the DNA polymerase copies DNA, it's 1,000 nucleotides a second, per gene a second. All right, so that's a physical performance that we aspire to from a technical perspective. Stated differently, I could imagine a future DNA printer that takes a genome, 12 megabases in it, yeast chromosome equivalent in it. And we're far from that. It's not this type of stimulated multi-gene system. It's a kind of computer program in 60%. It takes a long time. Did you have this current question?
Yeah. What group sponsored biology? What sponsored in
Bioengineering, which is stuff grows. Fireflies. See a firefly? I don't know if they're out here. It doesn't necessarily feel less tired. They blink. Insects and fireflies blink. That was so neat. There we're starting about seed, and how the poppies. Anybody broke out the poppies? Like this tiny little seed of poppies grows into this plant. The leaves are self-assembly solar panels that are cycling this. Right? Then you get this beautiful flower, and then the flower, then you see the seed product pop up, and it dries out and it explodes. It's a reproducing machine. And anybody knows that it's the most sophisticated natural technology anybody's ever seen. This bottom up. So nanotechnology actually works. The writing of the programmable nanoassembly puts atoms in the right place. So they're like cluster fireflies. But as soon as you like stop and think about what's physically happening in the calculated system, it's amazing. All right. And we'll get further days of speakers, so I should.
Thank you, Dr. Endy. We need to get our next speaker set up.
Sure.
That's a great metaphor for machine learning. In order to train a machine to recognize something you care about, you need a whole lot of what we call training instances. So you can't just go up to a machine and say, hey, recognize coral bleaching. You have to show it many instances of what a coral bleaching is. And then once it's learned enough, metaphorically, once the dog has sniffed the tube enough, then you can show your software a bunch of pictures of coral, and it will say there, 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 and there. Now, how does a dog do this? I don't know. But we can speculate. We can think about what a neuron does, a nerve cell. That's not a real neuron. That's a cartoon of a neuron. It's outwardly binary. That means it's either firing or not. And if it's firing, it's doing about the same thing every time. So we think of it as either on or off, because I'm a computer nerd. That's why I think of everything. What determines if it's on or off is what's going on the input ends. So all those little dendrite tips are connected either to sensing devices or to the outputs of other neurons. So if you've got trillions of these, you can wire them up in very complicated ways, and that's how you get learning dogs or learning people. So an artificial neuron looks much the same. So here I've sort of deliberately put the pictures on the left to suggest our sense organs. And so this is one that's connected, it's taking its input not from other neurons, but from some sort of metaphorical sense instance. And something in the middle there, made of software, is thinking about the inputs and either driving a 1 or a 0 to the output. Well, is this realistic? Yeah, actually it is. Imagine that this thing is a very, very, very primitive few-celled organism swimming through the ocean. And imagine it's trying to evolve an early eye. And suppose the algorithm in the bubble in the middle is the sum of my inputs 4 or more. In other words, have 4 or more of my inputs fired. Well, if what's making inputs fire is a change in the amount of light coming in, because a shark is about to eat you, and if the shark is close enough to make 4 neurons fire, make 4 inputs fire, then the output fires. Is this evolutionarily advantageous? It sure is, if that output nerve is maybe going to make your little flagellant tail twitch and get away from the shark. That's a survival technique if ever I've heard one. So here we can start thinking about maybe this is how eyes evolve. And that's just a single one. And what we're doing in modern technology is wiring together millions or hundreds of millions of them. Looking closer at one of these, if you've taken a week of Python, you can probably make it. The output is 1 or 0. It's a function of these parameters, the blue W's and this thing called bias. One way to think of how this works is any input that's on, you take its number, its weight, we call it. Out of all the weights, everything that's on, add the bias. If that's greater than 0, congratulations to nerd fires. How long would it take to write that in software? So people invented this. Actually, people invented this, I'm not going to say before computers, but before we started figuring out that you can do whatever you want with computers. So in the 1940s and 50s, people figured out some deep stuff. How surprising is this? Brains are made of neurons. Brains think. If we can make fake neurons, maybe we can fake think. And this brings us to the Markov perceptron. I love that thing. It's a perceptron. This is one neuron. This is just one. And built around 2.6 million then is 26 million today. So they didn't get very much good out of it. I do want to show you my favorite picture of science in 1958. This is Dr. Frank Rosenblatt, basically doing brain surgery. And I don't know if you can tell what's in that circle. It's a wire cutter. He's debugging his hardware with a wire cutter. So respect to him. But not much happened there. And by the 1960s, people had invented what you would recognize as computers and realized, well, let's not build that room-filling perceptron. Let's just simulate these on computers and we'll see what we can do. There were interesting results, even with a single neuron, if it had a whole bunch of inputs. There was theory about what you could do with multiple neurons, but computers weren't powerful enough. And then in 1969, these guys went and ruined everything. They proved rigorously that there are some things you just can't do with this technology. And so everybody got sad or they went into other technology. And not much happened for 20 years. No. Well, no, that's interesting. You've been patient enough. I'm going to fiddle with this for 1 minute. And if I can't get that to work, then I'm just going to talk no slides. It'll be a different talk, but you won't be sitting any longer in your chairs. Here we go. Brings us to the 1980s. People thought, let's commercialize the stuff. Let's make money. They tried. Yeah, nobody really made very much money off of it. They do stuff like handwriting recognition. That was pretty cool, but that was about it. And the technology went into hibernation for about another 20 years. Brings us to the current century. Computers caught up, and they're all tremendous successes. In the interest of time, I was going to show you how you wire up this technology to recognize a feature in an image. I think that's pretty interesting, but in the interest of time, we're going to skip it. I'm happy to, maybe afterwards, those of you who want to mill around after our third speaker, we can just fire up my machine and I can show you. But I guess I'm going to have to ask you to take my word for it, that what we can now do is have, for example, a network with many, many inputs. I only drew a few on the left, but there are many, many inputs. And each input is connected to a single pixel from an image. And then what happens in the intervening transformation is a mystery. The same way that you can't look inside your friend's head and figure out why it is that when they see a picture of you, they recognize you. Same thing, it isn't totally clear why they recognize what they recognize, but they do. And that will be the extent of my explanation of this technology. So I'm just going to skip that one and skip that one and that one and that one. Wow. No problem. I'm going to skip to the interesting part. The interesting part is, what do I do with this? Don't know. All right, you see my cursor. We're just going to simplify this. There and there. I'm going to tell you what I came here to tell you about, which is what I do with this stuff, because technology has no conscience. So when you hear of a really cool technology, many people will say, well, we're in Silicon Valley, let's go make a whole lot of money out of it. There are much more satisfying things to do with your life than figure out how to turn ideas into money. Turning ideas into making a better world, that's the most satisfying life. So here are two ways that I like to do it. One is called peripheral vision. I didn't misspell that. The phylum that contains all the sponges is porifera. So get it? See what I did there? And then oral vision, which is a much more difficult problem. So peripheral vision is the study of glass sponges. They're beautiful. That's not why I study them. They keep us alive. They can be quite big. I visited some that have been about this big, and I could fit in them. And the reason they're called glass sponges is that they filter silicon out of the water. And they use that silicon to build skeletons. And the cool thing about that is that if some living organism dies, if you come along 3 million years later, you've got no prior evidence of it. It left no trace. But when one of these dies, that glass is pretty much eternal. And we can study, for example, one thing I study is what happened the last time the oceans were climate changing to the degree that they are now. They did it once, and the sponges did something really interesting. But I'm getting a little ahead of myself. I want to explain to you why it is that these sponges are so ecologically important. They're important because they do a ton of processing. They're hotspots of ocean metabolism. So the entire food web can depend on them. I think many people know that ocean food webs spring abundantly when there are healthy coral reefs. But it turns out that when there are healthy sponge communities, that happens too. It's just that sponges don't have quite as good oppressive market. So they're not quite as popular. You can squirt green dye into the bottom of a sponge, and within seconds, it's pouring out the top. Let me not tell you, let me show you.
Small as bacteria, as well as larger particles. They might not look like they're doing much, but a simple demonstration shows how effective sponges on water. Where this rich fluid puts it on fluorescent. By squirting around the base of some sponges, we can observe how water is moving by much more than dye does. the guys come through the sponges a lot more. See the sponges really water pump and also a big straight. Right? Tube sponges are even more spectacular than research. They pump the place so furious that they look at the collection. Just so steps.
Well, I think I made my point. So there's a lot going on in the sponge. And ever since I any scuba divers in the room? No? How about not yet? It's an amazing thing to do. Since I got into sponge science, I've gotten in the habit when I'm flying over one of these sponges, I put my hand on top of it really close, and you can feel a little bit of jitter coming out on your hand. You can actually feel this going on. So these sponges are actively taking care of a large fraction of the ocean biome. And their skeletons are these beautiful biosynthetic glass things. And when the sea temperatures rise, as they are doing, protects the corals. Corals are crazy important. Bet on the sponges. The sponges, we are pretty sure, do a lot better. So this is the Bering Sea. On the right, you've got Alaska. On the left, you've got Russia. And my partner, who's a whole lot tougher than I am, goes up in there and drops horrors down on the bottom of the ship and extracts core samples. And then what you've got is sort of a time machine. The stuff from the deepest part of the core sample is oldest, and it's just all these deposits that have been laid down. At the top is the newest, and in the middle is the early Pliocene warm period, 3 million years ago, the last time the planet formed the way it's doing now. Sure would be a good idea to find out who the winners and losers are in those times. So I really think, because I love corals, we ought to protect corals. But then also maybe we ought to be seeding sponges to take care of the ocean until we get things under control and have the corals thrive again. What kind of sponges? Boy, it would be good to know, wouldn't it? Turns out that the spicules, the shattered fragments of this stuff, tell you what kind of sponge. And so we have a project going on now where we're investigating, just looking at the different kinds of spicules. Well, how do you do that? This is a micro photograph. And the thing that Dave's pointing to, yeah, that's not what we're looking at. Sort of between the A and the B at an angle, you see that thing looks like in here, that's what we're looking for. So traditionally, how we do that is you take a little smear from your core sample and you put it at the bottom of the microscope. At the top, you plug in a graduate student with a notebook, and you just have them write down everything they see. Well, you can see the 100 micron bar. And if you think of the core as bigger than salami, it's more like a bologna. So about that big around. So imagine what a tiny fraction of reality you're looking at. If you've got some poor grad student counting this stuff, and this is a grad student who hasn't had enough sleep and has other stuff to do, and is not an expert in the sense that a world expert is an expert. So this is going to be error prone. And what we want to do is scale this up. And the way we're going to do that is with one of these. It's a machine where you take your core sample stuff, mix it with some water, some other stuff, pour it in the top, and then drop comes down out the bottom, and every individual particle of anything is encased in a nice drop of water, falls past the camera, and you get an individual photograph, which we run through a machine learning classifier, which I would have explained to you, but interest of time, but I think you get the main idea. And that's starting to take off. So status, we got a proof of concept 2 years ago. We can classify is this from a sponge or not from a sponge. Pretty darn good accuracy. we now have a whole bunch more training data. Remember the dog with the sniffing stuff? The bigger your training data, the better your software. So we're developing that now, and we're also making it sophisticated so it can tell us what kind of sponge was in there. So now when we look at the various slices of bologna going back in time, we can find out how community makeup changed as the oceans got warmer. And this is some very valuable information indeed. So what? Well, the idea is to learn how these ancient sponges responded to warming seas. The processes of life, including the ocean, need safe spaces. The best safe spaces are corals and sponges. If the corals are going to be limping along for a while, let's protect them, but let's think about the sponges as well. So that's peripheral vision. The next project is coral vision. Because corals, well, 1/6 of the world's coastlines are fringed with corals. They support millions of species. One of them is us. I forget the fraction, but it's something like 1/10 of humanity gets their wild protein from the ocean. They are places of stunning beauty. And the best reason to learn how to scuba dive is to do this. More stunning beauty. Even more stunning beauty. And ecologically, these are called the rainforests of the ocean. These are the hot spots of biodiversity. They're hot spots of the various low-level processes of life, like nitrogen fixation and photosynthesis. If you don't know what those are, think of them as this is how neutral inert chemicals in the atmosphere turn into life stuff. Also, super important, they're shelters for juvenile fish. When fish, when they're born, are they're just super tiny, and so they're easy prey for predators. But corals, like sponges, have got nooks and crannies. They're like English muffins. And a tiny baby fish hiding in a nook and cranny will live to be a biologically important adult fish. Of course, they're different. I'm going to skip this part of the slide. I think you all get way too much information about the threat side of stuff. You don't need any more of it from me. Yeah, we'll skip that one. Ocean warming is bad. Pacific Ocean is bad. That's it. What do we do about it? Well, here we have a simple little device with a big pompous name. It's called an ARMS, Autonomous Reef Monitoring System. It's not a system. It's a bunch of 1 foot by 1 foot gray plastic plates all bolted together. It's autonomous in the sense that it sits there at the bottom of the ocean, and it doesn't need help from anyone to sit there at the bottom of the ocean. We just bolt it there. We leave it for about 18 months. That's called soaking it. So there's some probably bolting one down. Say goodbye to it for 18 months. Actually, in any experiment, you want to replicate your experiment, so you always put 3, not 1, in case of tragedy. And when it comes up, it's not a bunch of plastic; it's a habitat. And then by analyzing what's on each plate, you can figure out, this isn't going to tell you what was at the coral reef where you soaked your arms, but it is going to reflect the biodiversity and the general health. And this is the only cheap tool that we have for measuring health. So what do you do when you recover it? Well, the main goal was that you can scrape all the life off of it into a blender. Make a DNA smoothie, extract the DNA, identify the DNA, job done. Too bad that didn't work for certain technical reasons I won't go into. Boy, are we glad that before they did that, they took high quality photographs, because that's all we got now. Longitude, latitude, and a picture. Well, the other thing you can do if your DNA thing didn't work is just show the image or show the plate to a local expert. The local experts I know, marine biologists, you got to buy them a beer to get them to cooperate, but they will cooperate. Now the problem is that I've got 14,000 of these, and there's not enough beer in the world. So we're going to have to find another way. Noah, you heard of Noah? It's sort of like NASA of the ocean. And they did this huge, large-scale survey of pristine coral reefs in the Pacific Ocean. Too bad it was before I came on the scene. I would love to help them do that. But these days I'm working on the data. It was over 13 years. They deployed hundreds of units, thousands of places. I've got 13,000 images. So we have to scale up, and of course we're going to scale up with machine learning. I think this is a super cool project. Here's a life lesson for you. Why am I the one who gets to? I got the skill. It's not a crazy skill. It's just, a nice skill to have. I knocked on the door and I said, hey, I want to do this. And they said, hey, great. Because most of the people who have this skill are working for Google, Facebook, the big Silicon Valley companies, which I consider conscience neutral at best. The number of people who are willing to make a lot less money and have a lot more adventure, not so many. Do you want to make sub-Google salaries and have amazing adventures? This is open to you. And so Coral Vision is deep learning plus these photos. And the big ideas are to develop these neural networks that can analyze, just find everything on the place and then identify them. That's huge. We're a couple years in, it's going to take a couple more years. Then we want to empower the entire global research community. So what we want to do is create a website where anybody can do experiments on the data. They can also upload their own photographs because coastal high schools in places like La Jolla, San Diego, they use these in their marine biology classes. So we want anybody to use our analysis tools and develop their own hypotheses. What we have is 14,000 plates. Metadata, that's this word for the data that isn't the core data. So things like SST is sea surface temperature, pH is acidity, longitude, latitude, stuff like that. Step one is to find all the individual things. Now with the sponges, every individual particle went into its own drop of water and turned into its own photograph. So the hard part is done for us in hardware. Here, first, finding the individual things, it's called segmentation, is a much harder problem than identification. And I think that this way, classifying, like in peripheral vision, is this kind of white belt level. And identifying is more like, and segmenting is more like this. But we're starting to get results. Here's a sample photograph, and here's the analysis coming out. So we're starting to get coherent results. This is just in the past couple of months. So the community who cares about this is very excited about this result. And I'm going to stop there except to say that, and let's talk about the elephant in the room, which is, climate change. And my generation this is our legacy to you. But there are three possible futures for it. Of course, sorry kids, you're doomed is about the worst thing that could happen. I don't believe it. Not quite as good. I mean, just kidding, kids. Climate change is a hoax. Who was it who said that? And it's all good. Turns out we were wrong. Well, claims made without evidence may be dismissed without evidence. So why do I say that there's an outcome that's even better than you don't have to worry? That outcome is your future is in your hands. And not all of you are going to step up to that, but enough of you are. And our previous speaker has talked about ways to do it. I'm here to validate everything that he said. And with apologies for talking so fast and gratitude for your attention. I can either take some questions now or defer to the end. What do you think? Take it at the end? Sure. Yeah.
Our next and final speaker is Jocasta Manasseh-Lewis. She is a master's student in biology at Stanford University who is currently studying neurodegenerative disorders using computational models to identify biomedical phenotypes in order to improve their mechanistic understanding of the disease. She graduated from MIT in Stanford with a training in medicine.
I've never really thought about how curiosity, you guys know anything about universities? When they consider not having to that's fine. So the name system is a secondary they name the job, right? That's T cells, B cells, right? Antibodies. I think they're specific to action. And then immune system is very long. So generally you get a transformation or that renew that's the main reason. Right? How do you learn? Right? How do you adapt to situations? Your brain has to change. And that is one step. Yeah, so Alzheimer's disease is actually classified as 5.8 million people. And the number of people that suffer purple. So this is a massive public health problem. We want that we have a solution for. It was described as an 1800s, and people were looking at more biological kinds of causes in that. But as of now, we don't really understand everything about it or how to know about it. It makes up the majority of the country. And most popular symptoms are shown primary events. Often with Alzheimer's, don't forget there's working for it, key for the car, right? And this can progress to really floating poverty decline. So what do you think about the risk factors for Alzheimer's? Who might cause more? Who should have the symptoms? Sure. Yeah, that could be contributing factors and environmental factors, right? Anything else? Yeah, genetic. That's what we're going to talk about today. So, one of the risk genes, the one I want to highlight is ApoE. Specifically, you guys know what alleles are? The over genes. It's not, right? So alleles, right? You can have different sort of flavors and different effects, right? So the E4 allele is a massive risk factor. Right? It's actually the biggest genetic risk factor in most Alzheimer's disease. Hereditary is also a contributing factor, and persistent or chronic infection in diabetes. So you guys let me know in about 15 minutes. So another kind of, you're looking at someone's brain, right? MRI. You want to look at the patient's brain, right? You want to see what kind of things, MRI or imaging modality that would tell you some problems about this. So one of the most important ones is amyloid plaque seizure. This is process you can see for the brain. But one of the most important things is synapses. So, do you guys know the synapses? And we'll shout it out. Yeah, sure, in fact. Yeah, exactly. You're not communicating synapses. So, you have the presynaptic neuron, which is sending a signal, and you have the postsynaptic neuron, which is part of the body, receiving the signal, right? So, these synapses, right, are really important for plasticity. This is where a lot of plasticity happens, because you can change the amount that the postsynaptic neuron responds to the presynaptic neuron. This changes the amount of information that transfers the synapse. And it's by modulating this type of synapse up and down that you have learned. Right, so there are kind of two flavors of Alzheimer's disease. One of them is early onset Alzheimer's, which occurs obviously earlier in life. We look forward to persistent life. And there are three really high risk genes: APP, PSM1, and PSM2. And we can see that APP is a little bit lower in the scale, but it accounts for the majority of cases of late onset Alzheimer's. So this is kind of a surveillance hypothesis. If I now improve amyloid beta protein, amyloid plaques. Yeah. So basically, people have observed these plaques in Alzheimer's patients. So because you observe this, you can observe the situation, right? So basically, the APP protein is cleaved into the beta. We want to have lots of different downstream effects. And it's believed that this is the primary source of the effect, right? So here's a question. We think that a primary source of Alzheimer's disease. Why don't we have fear for it? Sorry, this is a little bit of a trick question. Sorry. It's probably because it's not a major cause, or not. It is a major cause, but it might not be the single contributing factor. Now, these are amyloid beta plaques, and what they're doing in this person's brain is they're surrounding protein spikes. What what effect can this have? What's the plaque, right? It's a very hard brain curable. It's surrounding the virus. And it's protective, right? Maybe it's protecting the brain from the virus. People think that maybe when you have a brain infection, right? So this is uncommon, right? So a blood-brain barrier. So your brain doesn't exchange blood with the rest of your body like the organs does, right? There's a very tight control, which is why it's hard to get therapy to brain disorders. But sometimes you can get infections and process that when you do, because your immune system in the brain is very different from the rest of the body. You don't have that blood issue. You can see these to protect against infection. And furthermore, when you have all of these plaques, it use falls on the downstream inflammation, all downstream effects, and this is just going to run away without having infection. Here's a little bit of context. Plaques aren't always the most toxic things in the brain. They're not always the most toxic version of the These can actually form smaller fibrils or smaller aggregates that are a little bit more neurotoxic. Another thing is that different cell types, different cell types. It's important in your constant brain regulatory to make sure that's going to cause or biological changes that happen. That does not apply. People use mouse models a lot to study human disease, but sometimes well, they're not perfect, right? I don't know if you've heard the phrase "all models are wrong, but they're useful," right? So maybe it's a useful model, maybe it's not, but equally in mice, and not so equally in humans. And again, talk about the fact that a wheel equally have does matter. Okay, so this is kind of a picture of, I guess, how equally is perceived in literature. So there's some kind of observation that maybe this has a beneficial effect. Maybe this stops inflation. And then there's kind of also the view that maybe this has a generative effect, that this causes changes of seen outcomes. And I think this is kind of a story with a beginning, the middle, but no end so far, and both can be true. So both are hoped. So another motivating factors, I don't know if there's been some of these meetings recently, but there has been a Alzheimer's drug, which is a monoclonal antibody against COVID-19. But there's been one made that is against amyloid beta protein, right? So this is basically stopping the aggregates from having downstream effects by binding to them and reducing the target so you can destroy them, right? But kind of the issue with this is that it's controversial, but it got kind of fast track approved by the FDA and had very low efficacy and thought of decline as well as Alzheimer's drug. So it kind of suggests that maybe targeting A beta doesn't fix the situation completely. Maybe it's a little bit higher effect. And so what we're going to talk about for the rest of this is about inflammation. So kind of the main role of inflammation, the classical role of inflammation is there's a cholesterol barrier, right? It transfers cholesterol into neurons. So the brain has very high energy requirements, and one of the nutrients that is really depends on cholesterol. Okay. So cholesterol is really important for membrane-dependent processes, right? You need cholesterol in the membrane when you're doing things like adding receptors, taking from the way, we talk a bit about CRISPR, right? So you can do things with CRISPR like knock out genes, so remove genes from the protein, right? So if you make mutations that cause the stop kill happening very early, you get a protein that doesn't really do anything, right? These are called not gene knockouts. Mice that are knockouts are equally viable, but they have a lot of as we expect, this is the major body area of large issues. We have some walls, there's some dysfunction, but also kind of going back to maybe these beneficial or not beneficial. So we talked before about this kind of early version of Alzheimer's, so we have mice, one mice, and these are kind of monotherapy. When you antibody is have incomplete, where it's kind of similar to drug, you do see some increased performance, increased ability, but it doesn't happen backwards. And the hippocampus is really important part of the memory, and then you see a lot of degradation of hippocampus and Alzheimer's. So this actually has an immortal story. So any of my R's and alpha-R's are two really important receptors for neuroplasticity. And typically, we want a lot of alpha-R's at the membrane, we want a strong synapse, and we need NMARs to facilitate this. Even before, for example, the allele that really predisposes you to Alzheimer's, that allele depletes these receptors at the membrane. And it suggests that if you carry the allele, it's just harder to make stronger synapse. And now we're going to talk about if we're going to look different concepts of mood function. So, two really important systems on the immune system are complement and recognition. So complement kind of is seen for physical function. Complement antibodies, complement macrophages, it kind of enhances the ability of the immune system to react to the threat. Basically, are running macrophages. Do you guys know what macrophages are? So macrophage is a big. Macrophages are in every tissue, they control the tissue, and they eat things that are possibly dangerous. Right? So most of the time it's good, right? Microglia, they can even eat A beta, right? So we talked about A beta is bad, right? A beta degrades synapses, A beta is neurotoxic. So yeah, you do want these certain macrophages to scavenge them. But what happens when you go right? You start eating your own. So sometimes it looks like ApoE again has its hands in that process. ApoE can facilitate information consisting into a kind of road phenotype. They actually eat synapses. The complement is activated. So that's what happens, right? When you have an infection. And not only that, infections can kill neurons. So when you have these neurons that are dead, you send these signals to cytokines. And this causes the glitch with cardiac phenotype for a certain synapse. So it looks like maybe ApoE has some effect where it causes microglia to grow. But and goes up to that spider, right? It looks like it also has some beneficial effects. It can inhibit complement. When you inhibit complement, no longer are we use like signals or complement going to grow. So it looks like it's an effect. Really not, but it actually be tested. Or basically, and energy, so upregulated estrogen, right? That is also upregulated belief. That leads to more growth. Hormone growth was not that. Finally, what happens? So carbon unfolds in release, right? Because one of the keystone pathogens for fusion develops. So this is bacteria that produces a lot of enzymes proteins. Some of this does to basically put up nutrients so that we eat them. And some of these will disturb your immune system. I think you can attack cytokines. It just has a effect of destroying markers in the body. These are the so one of the magic is that it actually will cut ApoE fragments. Will you obviously cut protein fragments? You think that protein is so valuable that it's sort of thing that it used to? Yeah. Once you cut ApoE, it probably isn't able to function as a control transporter, right? and how not able to regulate the immune system, right? You basically lost control of the system. Hopefully, you can also treat A beta. Remember, what A beta having a neurodegenerative effect. And not only that, but this is the FC education. I believe what we can see is that in AD patients, you have more sort of bars, so each bar is a dosage bar. You have more fragments, you can cut these control patients on the right, they have less fragments, so you have less pieces of fragments in the heat. So it looks like something is destroying the ingredient in the patients. And E4 has two arguments. One is the argument, and it's a big argument. And so it looks like E4 is really sensitive to degradation. The neurodegenerative, which is much less common in E2, is not at all sensitive, it's very robust against degeneration. So it also means people with E4 are just more likely to have a gap in the size previous. Then also, just now with the function similar, about your inflation is very toxic. You can basically situate these proteins which causes these athletes to have an immune system. And not only that, but mice, where amazing fusion develops, they can also develop AD-like cognitive decline. So it looks like causing this specific infection is enough to cause changes that are similar to AD. Isn't all bad. Actually, when patients wait with E4, the vitamin D levels seem to be really important. The more vitamin D these patients have, with E4, the less likely they are to have cognitive decline. Basically, when you have an infection, this can just start a reduction of the active forms of vitamin D. And the less vitamin D you have is kind of a marker of how the probably have higher infectious immune system is intolerant, right? So that looks like maybe much of the immune system too. So often what I really want to understand was how many people we want to understand these companies with all of these factors that are differently open E2, 3, and then there's the context, right? Does the person have an infection, right? They have an infection. That could, that you might have metastasises, right? You might have microglia, you might have common mechanisms. Right? This can bring up all of these different changes into the brain that can help explain why we see it we do so many different things, because it's just responding to its effect. So actually, around these plaques, you see kind of two different phenotypes. One is the cells in the brain called astrocytes, and one of those astrocytes like cholesterol neurons, right? And these cell type produces a lot of ApoE, especially. But the problem is around these A-beta plaques, it stops producing as much ApoE. It can stop supplying cholesterol to the neurons. And think about that. Think about that. What might happen if you stop applying cholesterol to the neurons? If you need it to build synapses, if you need it to kind of adapt to the environment, what happens to your cholesterol supply as well? Sure. They're not degrade, but it might be less effective, right? It might be less effective doing its job, right? It might be less adaptable, right? It looks like this cholesterol supply is really important to adapt when you have an injury. Right? When people have a brain injury, sometimes they can recover function, right? And that depends whether or not they can reorganize the neural networks in the brain to basically recover, right? And if you don't have the raw materials to build the network, I wasn't able to predict something, but neurons have to build a lot of surface area. Most cells in the network are very spherical, but neurons have projection, but there's a massive interchange, just like very long axons. So it's really, really important to neurons to have cholesterol because they have such complex mechanisms. So if you think about that, APOE's role as a cultural principle is really important. So, and talk about how microglia, if this is making microglia go wrong, right? It can make them start eating synapses. Right? What happens when you start eating synapse? Like what happens to the information of that synapse? It's lost, right? It's gone. And that's usually what's happening. You see synapse loss in mutations. Not only that, but the synapse loss actually seems to correlate better with cognitive decline than A beta does. Right? A beta is actually not a strong predictor of cognitive decline. So which do you think is probably positive, right? The one that correlates really well, or the one that doesn't, the one that is present but doesn't correlate well at all. In fact, you can see a beta plaques in autoimmune patients who do not have AD at all, which is people who score well on like a psychological exam that they're cognitively they're very normal, but you do a brain scan and they have lots of beta plaques, right? If these are beta plaques, they're really neurotoxic. When you're talking people to have cognitive decline as well, we just have them score relatively well and have like some psychological exam, right? So it looks like maybe A beta isn't the main thing that's causing Alzheimer's. It's definitely there, right? Definitely associated. It's definitely has a neurotoxic amount of plaques, after specifically. That's the third one. It's believed that maybe these plaques that act as reservoirs, maybe once you have them, it's like hard, durable plaques, and they're just kind of sitting there. You don't have effects, right? They can't go and modulate the synapse. One little piece of these plaques very well. Then they can go and you might biologically absent, right? You get A beta at a synapse and modulate and say, oh, you're A beta. And then that will have some biological effect. And really the causes of the change causes to want the synapse to be sitting there. Right? So basically what I'm trying to impress is that this kind of seems to be really, really important in Alzheimer's disease, right? We can get about lots of different things, not just A beta, but inflammation, right? Can we transition to this this tolerant phenotype? Can we make microglia do what it's supposed to do, right? Can we make them eat A beta, right? Can we make them stop these synapses, right? And maybe if we can do that, that would prevent the neurodegeneration. So again, this is the story with the beginning and the middle, but there's no end. Maybe one of you will find the end. Maybe we'll figure out how to use the 21st century. Yeah, that's all I have today. Thank you so much.
Thank you again, Jocasta. Do we have any questions for either of the two presentations that we just heard? If there are none, then I think that will conclude our speaker series. We would like to thank all the people who took the time out of their busy schedules to come and speak with us. This event would also not be possible without the support and guidance of our cover advisor, Ms. Lenz. Everyone knows something good about the bioengineering. New techniques such as genetic engineering and machine learning applications